In this section, we'll be focused on the chemistry of hormones. All the hormones fall into three chemical classes. There are steroids, peptides and glycoproteins, and then the monoamines. All steroids are derived from cholesterol, which has this four-ring structure, and they're secreted by the gonads and adrenal glands. The steroid hormones include estrogens, progesterone, testosterone, cortisol, corticosterone, aldosterone, DHEA, and calcitriol. The monoamines are derived from amino acids, so they're also relatively small. They're secreted by the adrenal glands, the pineal, and the thyroid glands. They include epinephrine, norepinephrine, melatonin, and thyroid hormone. Now, the peptides and glycoproteins are much larger hormones. They're created from chains of amino acids. Thus, they are produced by transcription and translation of genes that are located in the DNA. They're secreted by the pituitary and hypothalamus. Oxytocin, antidiuretic hormone, all the release and inhibiting hormones, and the anterior pituitary hormones are all peptide and glycoprotein hormones. This figure shows us the synthesis of steroid hormones. Here's the original cholesterol molecule, and it will be turned into progesterone, which in turn can produce testosterone, cortisol, or aldosterone. Something that's interesting to note here is testosterone is converted by an enzyme into estradiol, which is one of the primary estrogens. So if you think about it, if there's an excess of testosterone, there's probably also an excess of estrogens. The peptides are synthesized the same way as any other protein, through transcription and translation. The first form is an inactive pre-pro hormone. The first several amino acids is usually a signal peptide that guides it into the cisterna of the rough endoplasmic reticulum. Remember, proteins that are made for export from the cell will be made by ribosomes on the rough ER, and they'll be incorporated into the ER, which will then ship out the modified proteins to the Golgi apparatus, which then ships outside of the cell. Once the signal peptide is removed from the pre-pro hormone, it's called a prohormone. The Golgi apparatus does the final transformation and labeling for shipping to make it into an actual hormone that's packaged for secretion. Let's look at insulin as an example of this hormone processing. It begins as pre-pro insulin when it's first transcribed and translated and has some big disulfide bridges that hold these chains together. When we remove this connecting peptide, or C-peptide, in order to form the fully functional insulin molecule that you can see here, it looks as if this hormone is two chains. However, originally it was one gene and thus one long chain. It's the post-translational processing that gives it its final form as it moves through the endoplasmic reticulum into the Golgi apparatus. Let's look a little more closely at the monoamines. Melatonin is synthesized from the amino acid tryptophan, and the other monoamines come from the amino acid tyrosine. Thyroid hormone is composed of two tyrosine with iodines attached. The follicular cells of the thyroid gland absorb iodine ions. They transport them over into the lumen of the follicle as a reactive form of iodide, indicated by the star here. Inside the lumen, we'll see that there's a lot of thyroglobulin, very large proteins. This forms a colloid and contains lots of tyrosine. You'll see that the tyrosine and the iodine combine to form thyroxine, and the thyroxine remains stored in the lumen of the follicle, bound to thyroglobulin. When TSH is released from the anterior pituitary, it stimulates the follicular cells to remove T4 from the thyroglobulin and take it up into the cells. It releases these hormones into the plasma. Most of the T3 is produced in the liver or by the target cells that actually have receptors for it by removing one iodine from T4. 95% of our circulating thyroid hormone should be T4, and only about 5% is T3. However, T3 is the most active form, so we convert T4 to T3 at many of the target tissues. Most monoamines and peptides are hydrophilic hormones. That is, they enjoy being surrounded by water. And the steroids and thyroid hormone are hydrophobic. This hydrophilicity or hydrophobicity has an influence on how the hormones can be transported in the blood. Those that are hydrophilic will mix easily with the blood plasma, and those that are hydrophobic won't. So the hydrophobic hormones will find bound to transport proteins like albumins and globulins that are synthesized by the liver. The bound hormones have a longer half-life because they're protected from the enzymes of the liver and from kidney filtration by their association with these large globular albumins and proteins. However, it's only unbound hormones that can leave the capillaries to actually reach the target cell. Thyroid hormone binds to 3 different transport proteins in the plasma. There's albumin, thyrotin, and TGB, thyroxine binding globulin. More than 99% of the circulating thyroid hormone is protein bound. Steroid hormones will bind to globulins, like transcortin is used for the transport of cortisol. Aldosterone has a very short half-life. 85% of it is unbound, so it's easily accessible to enzymes that are produced by the liver, and it's easily filtered out. 15% of it binds weakly to albumin and other binding proteins. So as I've already introduced, hormones will stimulate only those cells that have receptors for that specific hormone. The receptors are protein or glycoprotein molecules that are embedded in the plasma membrane or free-floating in the cytoplasm or in the nucleus. Now, it depends whether you are a hydrophobic hormone or a hydrophilic hormone, whether you can actually reach receptors inside the cell. So those hormones that are steroid hormones can easily pass through the plasma membrane of the cell, and thus they'll bind to receptors inside the nucleus that are directly associated with the DNA. Receptors act like switches. They turn on metabolic pathways once the hormone binds to them. Usually, each target cell has a few thousand receptors for a given hormone, and the receptor-hormone interactions exhibit very tight specificity. That is, there's a specific receptor for each hormone. These receptors become saturated when all of the thousands of receptor molecules are occupied by hormone molecules. The hydrophobic hormones, as I introduced, will penetrate the plasma membrane and enter the nucleus of the cell. They act directly on the genes, changing the target cell physiology. Estrogen, progesterone, and thyroid hormone all act on nuclear receptors, and they take several hours to days to show their effect due to the lag time necessary for proteins to be synthesized and change the physiology of the cell. Hydrophilic hormones cannot penetrate into the target cell, so they must stimulate the physiology changes indirectly by binding to a receptor that's located in the cell membrane. Then we'll see activation of a multitude of different second messenger systems in order for these hormones to elicit their effect on their target cells. So let's take a closer look at thyroid hormone. It enters the target cell by diffusion, mostly as T4. It's released from its binding globulins in the blood and moves out of the capillaries through the cell membranes and into the nucleus. Within the target cell, T4 is converted into the more potent form, T3. And it's primarily T3 that will bind to the receptors on the DNA. T3 then activates the genes. It's one of the activating proteins that helps turn on the whole event of transcription of DNA. It might make muscle proteins like myosin, which enhances cardiac muscle response to the sympathetic stimulation, or it could be involved in strengthening the heartbeat. Thyroid hormone has a very broad spectrum of effects depending on the cell type that it's activating. Now let's take a closer look at the peptides and catecholamines, which are hydrophilic. The hormones bind to the cell surface receptor, and that receptor is linked to a second messenger system on the other side of the membrane. It activates a G protein, and the G protein goes over and activates adenylate cyclase. The adenylate cyclase will then produce cyclic AMP. Cyclic AMP will either activate or inhibit enzymes within the cell. It'll turn on metabolic reactions that are involved in synthesis, secretion, or changing membrane potentials. We took a brief look into these second messenger systems when we looked at how some neurons signal each other. This is called a second messenger system because the first messenger is like the hormone that knocked on the door of the receptor, spied through the little hole and told the G protein on the inside what it needed to do. So the G protein ran and took the message to adenylate cyclase, which turns on this whole cascade of events. So the G protein coupled second messenger system, in this case, involves cyclic AMP. But there are multiple other kinds of second messenger systems. There's ones that involve diacylglyceride or inositol triphosphate. They work in very similar ways. The hormone binds to the receptor and activates the G protein. The G protein may then go over and activate diacylglycerol, which is formed from a phospholipid in the membrane. We may see also that G protein goes over and activates inositol triphosphate, another second messenger. Either way, these second messenger systems activate a multitude of different processes inside the cell so that the hormone binding to the outside can have its effect on the internal physiology. Why do we need these second messenger systems is a great question. The reason is because one hormone molecule can then trigger the synthesis of many products inside the cell. This makes the hormones extraordinarily potent. We see here that maybe one hormone activates several cyclic AMPs, and those activated enzymes, which then each of those activates several different enzymes, and each enzyme produces several of the end products that have a very large effect. So second messenger systems are very important in hormones having such a prolific effect. Take a moment here to diagram at least the cyclic AMP second messenger system. Become familiar with the hormone, the receptor, the G protein, and the cyclic AMP, or DAG, or inositol triphosphate. Each of those three molecules function in the same way. They go on and activate protein kinases. Protein kinases are proteins that activate enzymes, so this cascade ends up having a very large effect. Diagram this out now. And then we can discuss how we modulate a target cell's sensitivity. Target cells adjust their sensitivity by changing the number of receptors in the membrane. So very few receptors will pick up fewer hormone signals and have lesser of an effect. But if the cell starts to produce more receptors and push them out to the membrane, then we can increase the response of the same dose of hormones on that target cell. We call this process upregulation. Downregulation is the opposite. We reduce the number of receptors and thus create a less sensitive cell. This happens when we have long-term exposure to high hormone concentrations. So, for example, in the condition of stress, where cortisol is bombarding the system, the cells simply have to become less sensitive because they can't have hormones knocking on the door constantly, being annoying. But when one cell downregulates its receptors for a hormone, it may find a way to bind to other similar receptors on different target cells, or the hormone might be in such large quantity that it's transformed into a different hormone by enzymes that are around to do that. For example, when we consider the cascade of conversion of cholesterol into estrogen, if we have a huge amount of testosterone and the enzyme is there to convert testosterone into estrogen, we'll end up with estrogen, and it has some weird effects when it's in the wrong place. Conversely, if there's tons and tons of estrogen, then it might shift towards testosterone, and we'll see higher levels of testosterone than we might normally see, and it might have some strange effects. Most cells are sensitive to more than one type of hormone and exhibit some interactive effects. These effects could be synergistic, permissive, or antagonistic. In synergistic cases, we have multiple hormones acting together for a greater effect. We'll see synergism, for example, between FSH and testosterone on sperm production. Both of them are necessary for successful production. There are permissive effects where one hormone enhances the target organ's response to a second hormone. For example, estrogen prepares the uterus for the action of progesterone in maintaining the uterine lining. In antagonistic effects, we might see one hormone opposing the action of the other. We've looked at an example of that already when we considered insulin and glucagon. Insulin acts to lower blood glucose, and glucagon is acting to raise it. So now we've explored how we get a signal to the cell. How do we turn that signal off? It's necessary to turn off the signals when the hormones have served their purpose. Otherwise, we'll continue to produce too much of an end product. Most of the hormones will actually be taken up and degraded by the liver and kidney, and they'll be excreted in bile or urine. The metabolic clearance rate of a hormone is the rate of removal from the blood. It has a half-life, which is the time required for 50% of the hormone to clear from the blood. The faster the metabolic clearance rate, the shorter the half-life of the hormone.